In the previous part, we discussed some experimental results published in the PNAS, which led us to the following interpretations. The urinary output of modified bases is directly proportional to oxygen consumption. The rate of oxidative DNA damage is directly proportional to the rate of energy metabolism. And lifespan is inversely related to the rate of energy metabolism. The interpretations may appear very strange. However, during the times when the experiment that we discussed in the previous part were done, the experimental data were consistent with the two prevailing notions or concepts. One, that the DNA damage plays a crucial role in the process of aging, and second, larger animals live longer because they have lower specific metabolic rates and therefore lower rate of oxidative DNA damage. As long as cells are metabolizing, there will always be production of reactive oxygen species, and therefore the DNA is constantly under threat. Let us discuss now as to what determines the extent of DNA damage. There are two very important factors. One, persistence of DNA damage, and two, mitotic activity of the cells. Let us talk about the first one. Persistence of DNA damage means how long the lesion remains in the DNA without getting repaired. If the lesion hangs around for a long time, then there is an increased chance that this lesion may never get repaired and will result in getting permanently fixed in the newly synthesized daughter strand in the form of a mutation post-DNA replication. If the mutation compromises structural and functional integrity, it may contribute to aging, and hence the importance of an efficient DNA repair system. A faulty or an efficient DNA repair system will cause persistence of DNA damage followed by the consequences. The second one is mitotic activity. If the mitotic activity is very high, that is, if the turnover of the cells is very high, then the DNA repair machinery does not have sufficient time to repair the lesion caused under oxidative environment. And thus, there are chances that the lesion will be passed on to the daughter cells as a mutation. Thus, overall we can conclude that more the persistence of DNA damage or more the turnover of cells, greater will be the consequences of DNA damage, which includes aging. It's time now for two very important facts. The DNA repair system in humans is far more efficient than the mice, and the turnover of cells in humans is much lesser than that of mice. So can we say that humans live longer than mice because humans are better protected from the consequences of DNA damage? And why are they better protected from the consequences of DNA damage? This is because they have more efficient DNA repair mechanism, and the turnover of cells, that is the rate of cell division, is very low. Think about it until the next part, in which we will discuss some more experimental results.